on the SELs, we know that there are very interesting markers in terms of the imaging perspective. I would say the MRI perspective, because it's the way to ensure that there is an expansion. So the way the technique is done, so the technique is done by looking at not only one, let's say, one-off picture, because it's multiple images of the same person that are kind of, registered all together. And then we can be sure 100% that that specific area, which is the lesion, is expanding over time, because there is this specific technique looks at the deformation of that area. So if the deformation is positive, we say like a positive deformation, and this is quantitatively calculated with looking at these Jacobian values. We find out that it's quite of it's quite sure for us, because many times we face also the situation where an area, a lesion can disappear or can actually shrink. There can be changes that are different. So the fact to see that that lesion is still there and chronically expanding, so this is also called slowly expanding, is I would say also is a very sensitive marker on that side. And what we've done so far, I mean, the studies have been done since 2018, 2019, have been published also in large cohorts, so large trial cohorts of relapsing, remitting, primary progressive MS patients, and they found that those SELs had an impact, and they were something different also from the new lesions or the technique that usually is manual. So the fact that this automatic technique gives a lot of benefits in terms of having higher accuracy, higher sensitivity. Therefore, we extended that in another trial at UCL that was involving the secondary progressive MS. So those are people at a late or advanced stage of MS, but where it is supposed that there is a predominance of chronic active lesions. And we found that those markers were quite important. So the figures, the percentages of these lesions was very high in secondary progressive MS. So I think it's very promising in a way, and it gives us an idea that SELs could be used in the future as one of the main markers for the chronic active lesion. The association also with all the clinical scores in this last trial. I was part also of this trial as an assessor clinician, and there was a multitude also of measures we collected. So from cognitive tests up to, hand disability, walking speed tests. So all those measures were also in a way correlating with the SEL. So this is reassuring and gives us that it could depict in a way the disability. So it gives us an idea also of what is happening clinically. So not only in the images. But yeah, the thing I think that the next step is also to understand whether they could be useful then from a more practical perspective. So looking at their effect on treatments. So if a treatment is really, modified, not if the cells, let's say, are changing depending on the treatment. So if we can guess, let's say, that there is a response to the treatment or not. So I think this would be probably the next what we expect for the future to see. For sure, now the situation is, it's very complex, and MS is also a complex disease. So it's not easy to say these markers will be the only marker that we will use in the future for looking at the treatment response, and especially now that also I'm presenting this work on combination of other markers that are as well used in MRI, and they are very well established to have an impact. So I think that we will need many markers altogether, and the future is also to try to integrate them, to have like an integration of those markers and create, for example, a profile of a risk, let's say, and also of our response to the treatment. So having those markers that comes not only from the MRI and the imaging, let's say, research area, but also from biology. For example, neurofilaments are as well extremely important, and there are my colleagues who will at the MS Frontier discuss also this topic. And I think that the combination of the two is the answer for the next future.